Hi students, welcome to our EeeZee Biology. Today we are going to discuss about urea cycle. The urea cycle is the first metabolic cycle. The cycle to be discovered by Hans Krebs and Kurt Hans Elliott in 1932. So this urea cycle is known as Krebs-Hans Elliott cycle. Next, the urea cycle is otherwise called as Ornithine cycle. Why that is Ornithine cycle? The ornithine is the first member of the reaction. So that the cycle is called as Ornithine cycle. The urea is synthesized in the liver and transported to kidney for excretion in urine. Next, structure of urea. The structure of urea, NH2, C double bond O, NH2. Here combines two waste product into one molecules of carbon dioxide and another waste product is here ammonia. Next, that urea cycle. In urea cycle, totally 5 enzymes are involved. First enzyme is a carbamyl phosphate synthetase 1 and ornithine trans-carbamylase. These two enzymes are synthesized in mitochondria. Next, third enzyme is a arginosuccinate synthetase enzyme and arginosuccinate lyase enzyme and arginase enzyme. These three enzymes are synthesized in cytosol. Next, the urea cycle takes place totally 5 steps. Next, we will see the steps of urea cycle. First, step 1. Step 1, formation of carbamyl phosphate. Here, condensation of ammonia and carbon dioxide with the help of carbamyl phosphate synthetase 1 enzyme. Here, 2 ATP molecules are consumed. So, the 2 ATP molecules is converted to 2 ADP and phosphate, and 1 phosphate molecule is joined to this carbon dioxide ammonia, so that we form a product is a carbamyl phosphate. This is your first step. Next, second step. Second step, that ornithine combines with carbamyl phosphate to form citrulline. Here the enzyme ornithine trans-carbamylase enzyme, the product is a citrulline. This enzyme and this enzyme are synthesized in mitochondria. These two steps are takes place in mitochondria. Next, the citrulline is enters into cytosol. Here third steps. Here third steps. Again the condensation process. This citrulline combines with aspartate enzyme to form aginosuccinate. Here water molecules are removed from those amino groups 1 hydrogen molecules, and from those amino groups 1 hydrogen and here 1 oxygen. So totally H2O molecules is removed from this step and combined to form aginosuccinate. Next, fourth step. In fourth steps, formation of arginine. Here cleavage of the aginosuccinate with the help of aginosuccinate lyase enzyme. Here from aginosuccinate to removal of fumarate and arginine is formed. Next, fifth step is here formation of urea. Here water molecules are enters into this reaction with the help of arginase enzyme. The water molecules is split. OH and hydrogen molecules are here NH2 and C double bond O NH2. This is a urea, and remaining NH2 CH2 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 and CH NH2 COH. This is a ornithine and this is a urea, structure of urea. Here NH2 C double bond O NH2 is here structure of urea. In urea, C double bond O groups, that is coming from carbon dioxide groups. NH2, one nitrogen atom is from ammonia and another nitrogen atom is from aspartate amino acids. The final product ornithine is regenerated and enters into mitochondria for its reuse in the urea cycle. Next, we will see about the disposal of urea. Urea produced in liver and freely diffuses and is transported in blood to kidney and excreted in urine. A small amount of urea enters into intestine where it is broken down with the help of bacterial enzyme urease to form carbon dioxide and ammonia. This ammonia is lost in fecal matters or reabsorbed into blood. In renal failure, the blood urea level is elevated. That condition is called as uremia. So resulting in diffusion of more urea into intestine and its breakdown to ammonia. So here blood ammonia level is increased. That condition is called as hyperammonemia. That is commonly seen in kidney failure patients. Next we will see the disorders of urea cycle. Totally 5 disorders. First one is here hyperammonemia type 1. Here due to the deficiency of carbamyl phosphate synthetase 1 enzyme in mitochondria. So the features are high levels of ammonia in blood and urine. Second disorder is hyperammonemia type 2. Other name is ornithinemia. Here due to the deficiency of ornithine trans carbamylase enzyme, so result in elevated levels of ammonia in tissues. Third disorder is here citrullinemia. This is a rare disorder due to the deficiency of arginosuccinate synthetase enzyme. Here result blood level and urinary excretion of citrulline is increases. Next, fourth disorder arginosuccinic acid urea. Due to the deficiency of arginosuccinate lyase enzyme, here increased levels of arginosuccinate in blood. Next, fifth disorder is here hyperaginemia due to the deficiency of arginase enzyme, result in increased levels of arginine in blood and increased urine excretion. Common symptoms of urea cycle disorders includes elevated blood ammonia level, and some patients affect vomiting, mental disorders and coma. Finally, some patients affect death also. Thank you students. Thanks for watching this video.